Chapter 9, Energy Metabolism, Screencast 1. This is the first of 8 screencasts for Chapter 9. The first is an introduction to the concept of metabolism, reactions, and the role of nutrition. Now, metabolism refers to the chemical processes that are involved in the maintenance of life. Basically, they enable us to release energy from energy-yielding nutrients, carbohydrate, fat, protein, and alcohol, and also allow us to synthesize new substances and excrete waste products. A metabolic pathway is a group of related biochemical reactions that occur in a progression. When we talk about the term intermediates are compounds formed during these steps. A little bit later, we'll talk about the difference between intermediates and products. When we are talking about the oxidation of glucose in the presence of oxygen, the products are going to be CO2, water, and ATP. Those will be the only products, and anything that's not one of those we will consider to be an intermediate. Now, we can break down chemical reactions in two general categories, anabolic versus catabolic. Anabolic pathways are those that start off with small compounds or building blocks and build larger ones. So example, if we look at glucose, fatty acids, cholesterol, and amino acids, they are the building blocks, and when combined will make larger biological molecules. Anabolic pathways require energy, and unsurprisingly, they are most prominent during stages of growth. Catabolic pathways, on the other hand, are breaking compounds down. So example, glycogen is broken down to make glucose. This will result in the release of CO2, water, and energy. This is more prominent during weight loss or wasting disease, but keep in mind here the difference. Anabolic require energy, whereas catabolic will be releasing energy. Here we basically look at a number of the major types of anabolic and catabolic reactions here. The first, we're looking at carbohydrates, so we can see two glucose molecules with the addition of ATP are forming glycogen. The catabolic reaction will be breaking down glycogen, the release of ATP, and two glucose molecules. Here we look at lipids, so glycerol plus fatty acids, once again, with the input of ATP, will result in the formation of a triglyceride. And if we break a triglyceride down, we're basically going to look at the release of energy, and the products will be glycerol and fatty acids. If we look at the last anabolic reaction, we're talking about the formation of proteins. So amino acids combine with the input of ATP, resulting proteins. Proteins will be broken down into corresponding amino acids with the release of ATP. Now here we're basically looking at the three general stages of catabolism, and what I want you to take note here is notice where they all funnel in. They are all funneling into the citric acid cycle, and notice I'm going to put an emphasis on acetyl-CoA. When we look at the stages of digestion, the first, breakdown of complex molecules to their component building blocks. Proteins will be broken down to amino acids, carbohydrates to monosaccharides, lipids into fatty acids and glycerol. Alcohol will be broken down directly to acetyl-CoA. Conversion of building blocks to acetyl-CoA or other simple immediates, so notice everything pretty much funnels into the citric acid cycle and the electron transport chain as acetyl-CoA. The metabolism of acetyl-CoA to CO2 and formation of ATP is the third stage of catabolism. Now, you've heard me use the term ATP or adenosine triphosphate. We want to think of this as the cellular energy currency. It is the only energy that can be used directly to synthesize new compounds, conduct nerve impulses, contract muscles, pump ions across membranes. It is made up of adenosine bound to 3 phosphate groups, and those phosphate groups contain high energy. And it is the hydrolysis of ATP which actually results in the release of energy. Now here we're looking at a cycle as far as the formation and breakdown of ATP. Let's start first with the formation of ATP. The starting point is ADP, adenosine diphosphate. Energy is necessary to attach the phosphate group to make it ATP. And notice we have these high energy bonds in the phosphate groups. Then, basically, we're going to see ATP hydrolyzed. Energy is going to be released along with a phosphate. Now, the type of reactions that we will spend a fair amount of time focusing on are known as oxidation-reduction reactions. Now, ATP synthesis involves the exchange of ions in the form of hydrogen ions from energy-yielding compounds. Oxidation-reduction reactions are the type of reactions that are used in this process. Electrons are transferred eventually to oxygen, the formation of water, and the release of energy is used to produce ATP. Now, a substance is oxidized when it loses one or more electrons or hydrogen, or gains an oxygen. A substance is reduced when it gains one or more electrons or loses oxygen. Enzymes basically control these oxidation-reduction reactions. Something that may help you remember, okay, Leo, loss of electrons is oxidation. The lion says Gur, gaining electrons is reduction. But kind of tie in reduction. You're making something more negative, and that's what's happening when you gain a negatively charged particle. Now, the other thing that we want to address are two B-complex vitamins that are major players in energetics. Niacin and riboflavin, they help transfer hydrogens from energy-yielding compounds to oxygen and metabolic pathways. Niacin functions as coenzyme nicotinamide adenine dinucleotide, known as NAD. The oxidized form NAD found in cells. Reduced form NADH also found in cells. Riboflavin also plays a role very similar to that of niacin. When the oxidized form is flavin adenine dinucleotide, or FAD, the reduced form will be FADH2. Now, keep in mind, we're going to refer to NADH with the corresponding hydrogen ion, along with FADH2 as electron transport carriers. Notice they've been reduced, so they've become more negative because they are actually transporting negatively charged particles. Now, during exercise, lactate dehydrogenase reduces pyruvate to form lactate. Now, this is really important. So we are now looking at a process that takes place when oxygen is not available to produce ATP within the mitochondria. There's two hydrogens from NADH and hydrogen ion. These are gained. Lactate can actually be oxidized back to pyruvate. Two hydrogens are lost. NAD plus will be the hydrogen acceptor, and what we'll see later, this is a way of keeping the generation of ATP going. This will take place outside of the mitochondria, and generally we'll look at a total of 2 ATP that are produced anaerobically, and keep this in mind, 2 ATP are better than no ATP.